Well, first of all, many thanks, Peter, for this definitely over-generous introduction, but I thank you very much. Our friendship does indeed go back many years, and it's always a pleasure for me to come wherever you organize something, whether it is in Szeged or Budapest, because the meetings you design are always remarkably well organized. This is, in fact, the second time that I'm here at the National Academy of Scientist Education. So this is a picture from 2019 when I was here. And that occasion, I confessed, and I do that again today because obviously most of you were not present in 2019, that unlike many other people, I was not a great science enthusiast when I was at school. In fact, my main interests were in the arts, and my interests were principally in classical music and in literature. But I did know about Szeged very early on in my life, and that is illustrated here in the bottom right corner of the slide here. And it was actually, to be honest, not Albert St. Georgi that I was very conscious of at that time, but it was the very great classical conductor, Ferenc Fricsay. He was one of my very great heroes when I was a boy. He was an outstanding conductor, but he grew up here in Szeged, worked at the Opera House in Szeged, and actually in this very charming opera house that many of you, of course, are familiar with, you will find this plaque here, which is obviously written in Hungarian, that commemorates his tenure in Szeged and his, unfortunately, all too short life. He then went on to Budapest, and then through connection with the very great German conductor Otto Klemperer, had an enormous international career. Now, today I will do something that maybe is a bit unusual, but it is inspired a bit by a lecture I heard many years ago, actually, by the great British physiologist and Nobel laureate Alan Hodgkin, who, when he gave the Physiological Society's annual review prize lecture in 1976, it was a very charming lecture, and he started out by saying that he felt it was time to come clean about how at least his work had progressed. And he said there's something slightly dishonest, he said, about the scientific papers that we write. There's a kind of background, then there are the logical questions that follow on from that, then there are the logical results following from that, and then we reach the logical conclusion. And as he pointed out, that actually was not at all how they worked. There were many setbacks, there were many mistakes. They, for a long time, didn't actually know what they were doing. They were just making masses of recordings from squid axons. And at the end, nevertheless, emerged a fantastically successful theory, of course, and this very famous series of papers in the Journal of Physiology in '52, which many people still today regard perhaps as the most beautiful papers in physiological sciences. Now, I wrote an editorial in Function, a new journal that I edit for the American Physiological Society recently, in relation to increasing number of papers that are not correct. And I made the point that many of these errors actually never get corrected. So maybe other papers are published that to some extent said it right, but the wrong papers still continue to exist in the literature and can cause a lot of confusion. So I remember my shock when, as a medical student in Copenhagen, I was told by a very senior consultant physician that we should not be naive, but medical doctors actually kill a lot of people. So that was quite shocking for me as a medical student. And maybe today, for many of you, it may also be a bit shocking to realize that actually a very large number of papers possibly even a majority of published papers are plain wrong, and not just in interpretation, but actually results are straightforwardly wrong. So I will use part of my own experiences in illustrating this process of starting out with something that turns out not to be right, how to get it rectified. And I will give a few examples of how this has worked out. When I started my scientific work as an undergraduate medical student in Copenhagen without any supervisor, the knowledge that was in the field was basically contained in this book by Horace Davenport, very well known at that time physician and physiologist, and the American Physiological Society still continues to honor him, and I had the great honor of giving the Davenport lecture some years ago. Now, in this book, which somehow was a book that everyone sort of had as a starting point when you start out, there was also a short chapter on the salivary glands, and I happened to have, for various reasons, which I, for lack of time, will not go into, I started working on the salivary glands. So I don't want you to look too much at this, and I will pass over very quickly, because basically everything on this diagram is wrong, which is in a way quite remarkable. But absolutely everything that you see there is incorrect. Now, the most interesting observation for me, because I was an electrophysiologist, was this idea that secretion in the salivary glands, and by extension also in other exocrine glands, was driven by a direct acetylcholine activation of an electrogenic chloride pump. So what Anders Lundberg, who actually was a quite prominent neurophysiologist at that time, and who had worked with such a prominent figure as Sir John Eccles in Canberra, so he had substantial credentials. And the reasons that he thought that what he called the secretory potential was a direct result of activation of an ion pump rather than opening of ion channels was that he could not find a reversal potential for the secretory potential. To add to the further confusion, he, for some very strange reason, decided to do everything upside down. So these are actually hyperpolarizations, which we normally would display as a downward deflection, but he decided to do it the other way around. So by comparison with, for example, a typical inhibitory postsynaptic potential from John Eccles at that time in Canberra, where you have a very clear reversal potential. There was no reversal potential here, and you see this is an apparently very large span of membrane potentials. So that looked very convincing. And when I and my classmate, both of us without any supervisor, came to the conclusion that this was all wrong, and there were many details wrong that I don't have the time to go into. Of course, everybody in the institute in Copenhagen, which was quite critical actually, of course, were sort of dismissing it completely. How could two undergraduate students have dismissed what a well-known professor of neurophysiology in Sweden had shown? But we were right, and Lundberg was actually wrong. This became even more clear when we moved from salivary glands to the pancreas, because in the pancreas, unlike the salivary glands, the change in potential was not a hyperpolarization, but a depolarization. And quite early on, it also became clear that there was a very clear conductance change. We realized that technically what Lundberg had done, he worked with double-barreled microelectrodes, and these double-barrel microelectrodes have the inherent danger that the coupling resistance between the two barrels can change when current is passed through one of the long. So what had happened was actually that this apparent very large change in membrane potential that Lundberg thought he had achieved was almost nothing. So the membrane potential was basically not changed in those experiments. And of course in the pancreas it was somewhat technically easier because the morphology is very clear. And we discovered, and that was pretty important, that all ASNA cells in an ASNA unit are very strongly electrically coupled. So the whole of the lateral membrane is full of gap junctions. And we did actually a lot of work with Laureates in Geneva on the morphology of this. So we realized that we could actually use the whole Eisner unit, basically electrophysiologically, as one cell. And then we could actually very clearly demonstrate that there was a reversal potential, and that reversal potential essentially in the mouse pancreas seemed to coincide with the chloride equilibrium potential. So it seemed that certainly one important component of what had happened was actually opening of chloride channels. Only very much later were we able to sort of reach a conclusive stage with that, when we were able to combine the patch clamp whole cell recording technique with calcium measurements, calcium imaging, and on-caging of cage calcium in pre-selected localizations. And here you see the single Eisner cell that gave rise to all these recordings that we are shown here. So we had cage calcium in the cell, we had a calcium sensor, and we could then on-cage calcium here, here, here. And as you can see in the recordings here, it is only when we on-cage calcium near the apical membrane through which secretion occurs that we actually got the switch on of the chloride channels. So this was probably a fairly conclusive demonstration of the calcium activated chloride current. You can't do single channel recordings with this particular channel because the single channel conductance is very low, 1 to 2 picosiemens, so you don't get neat recordings. Long before that, we had, of course, done the first single channel recordings in epithelial cells. And here I was just very, very lucky. In the autumn of 1980, Erwin and Bert Schekman invited me to come to Max Planck in Göttingen and give a seminar, and of course I was very happy to do that. And that's another important lesson, actually. So they had just discovered the gigaseal, the famous paper by Sigvars and Neer, which was published in the autumn of 1980. And I just happened to arrive in Göttingen a few weeks after the publication of that paper. In fact, at that point, they already had all the different configurations of the patch clamp technique. And unlike what many people would have done at that stage, try to keep it for themselves, give themselves an advantage of working with this technology before kind of helping a lot of other people to compete with them, as it were. This was, they took a totally different approach. So when I came there were already masses of guests in the institute, and both Erwin and Bert showed everyone everything. There was not a publication at that point about the patch clamp amplifier. There were, of course, no commercially available patch clamp amplifiers at that time. But they shared all their knowledge, all the diagrams with everybody actually came. This, what some people might think was sort of not very clever, because then they created a number of groups who actually could do also very nice work. But actually, it turned out to be very advantageous, because it really gave an enormous boon to the whole field, and undoubtedly helped also to get them the very well-deserved Nobel Prize relatively early on. Now, these calcium-activated potassium channels that we identified in the pig pancreatic Eisner cell initially were definitely in the basolateral membrane, because in more intact acini, we took the membrane patch from the basal part of the cell. So we knew that they were present in that part. And you can see very clearly it's a voltage-gated channel. As you depolarize, the opening increases enormously, and as you increase the calcium concentration, you also see how the open state probability is markedly increased. And by combining knowledge from the isolated membrane patch in the inside-out configuration at different calcium concentration and doing the voltage open state probability measurements, we could then compare that with the situation in the intact cell. And it became an early and important measurement of calcium in these epithelial cells, and that quantification still is actually quite correct. I developed a particularly strong connection with Bert Schekman, who was a very frequent visitor to Liverpool in the 80s, and we always welcomed him with great enthusiasm, and he was immensely helpful to me personally also, for which I shall ever be very grateful. Now we could put these things together, and by doing wholesale recording and combining that with internal perfusion of the patch pipette, we could change the internal composition of the cell fluid. And you can see here acetylcholine switches on both the potassium current and the chloride current, and if there is calcium outside the cell, then these currents stay there forever, as long as acetylcholine hits the membrane. And this can be mimicked by dialyzing a high calcium solution into the cell interior. And here you see that just by elevating the calcium concentration, you also switch on these currents. You can also switch off the acetylcholine-evoked currents by putting in a calcium chelator into the cell. Then we stop the currents. And finally, it also illustrates that the initial calcium rise in these cells comes from internal stores. So unlike the situation in neurons, where most of the calcium is increased by activation of alpha-gated calcium channels, in these cells there is equally good switch on of the currents in the absence of calcium-free, but it is transient, it doesn't last. And only when you readmit calcium to the outside solution, then we switch on the currents again. So we finally had a reasonably complete model of fluid secretion with the calcium-activated potassium channel in the basolateral membrane. The tight junctions are sitting here, and it is the tight junction, of course, that separates membranes with very different composition and ion channel content. And of course, the part that actually consumes energy is the sodium-potassium ATPase, and that's where energy is required. In the case of the pancreas, unlike in the salivary glands, there is a hormonal input by CCK, cholecystokinin. And this is actually one of the few examples where endocrinology became a bit simpler. Originally, this hormone was called CCK-PZ, which stood for cholecystokinin pancreozymin, illustrating the two principal actions of the hormone, namely to contract the gallbladder and to activate enzyme secretion. But later on, it was decided to dispense with the pancreozymin, which from a pancreatic point of view seems a bit unfair, since we like to think that the CCK action of the pancreas is quite important. But we'll come back to that point in a moment. So, of course, with CCK, as with acetylcholine, we get both fluid and enzyme secretion. If we stimulate with secretin, which specifically activates duct cells, which is a system that Peter Hegge particularly has explored with enormous success, and I don't have time to deal with that here, but we did write a very enjoyable collaboration and a review together, which also contained quite a lot of detail about the duct process. But the ductal fluid secretion is principally a cyclic AMP-controlled phenomenon, whereas the CCK and acetylcholine-evoked fluid and enzyme secretion is principally a calcium-activated phenomenon. So, how do the calcium signals actually work? Of course, for very many years, after we had discovered that calcium release in both the pancreas and the salivary gland occurs by release from internal stores, there was always, at all calcium meetings, a discussion about what was the link between, for example, acetylcholine activation of receptors and the release of calcium from intracellular stores. Of course, a prerequisite for that kind of signaling is obviously the knowledge that there is calcium accumulating organelle in basically all nucleated cells, so not in the red blood cells, but in almost all other cells. And the one who, together actually with Hasselbalch, discovered this was Satsuro Ibashi, a scientist who I think did not get quite the recognition that he deserved. It was the insight of Mike Berridge in this single author paper published in Biochemical Journal in 1983 that pinpointed that IP3 was the signal molecule that most likely was releasing calcium from the internal store. Interestingly, Bob Michel, who had worked in this area for many years in the UK, had reached the conclusion that phosphatidylinositol bisphosphate was a substrate that was broken down by the activation of phospholipase C. And strangely enough, in the paper he published in 1981, there is not mentioning of the word inositol trisphosphate, which is very, very strange, because if you look at the chemistry of it, that should be an obvious conclusion. But Mike actually measured the time course of IP3 formation, showed that it is by far the earliest biochemical event after receptor activation, and that it actually occurs even quicker than the rise in the calcium concentration. And of course, later that same year, the famous Streep et al. Nature paper that actually demonstrated in permeabilized pancreatic Eisner cells that IP3 would release calcium from internal stores. Later on, we came into the molecular age with the cloning of the IP3 receptor, and of course, a lot of work subsequently was carried out. Now, at that point, we began to be interested in what do the calcium signals actually look like. It would seem somehow not very clever of the cell, if one can put it that way, that it should just release the whole of the endoplasmic reticulum and then later on having to reaccumulate the whole thing. It would be far more sensible if you just signaled with very small releases of calcium. So we thought initially we would approach this in an electrophysiological way, and we did that using, again, the patch clamp technique. And we monitored the calcium-activated chloride current as a way of monitoring the calcium changes at the point in the cell where it matters in relation to secretion control. And you see that with the active isomer, the 1,4,5 isomer of inositol trisphosphate, we got these repetitive spikes of calcium-activated chloride current, and we assumed and concluded in this paper that these must be local rises in the calcium concentration near the site of secretion control, and similar findings were made with acetylcholine. Now, very shortly after our paper was published, I had a sort of unpleasant experience. I came to a meeting in Göttingen, and Erwin Neher told me that work that he was not personally involved in, but some other people in the institute had actually tried to measure calcium changes by putting IP3 into the pipette. They didn't do the electrophysiology, they just measured calcium. And he said, actually, surprisingly enough, in view of this result, they didn't see anything. Only when they stimulated with very, very high concentrations. So I was told that most likely this was wrong. Now, initially, it looked as if they might be right, actually. So, of course, we went back. We were just at that point beginning to be able to measure calcium in our cells, but at that point, we could not do imaging. So this is photon counting. And indeed, with the low concentrations of acetylcholine that gave rise to these repetitive spikes of calcium-activated chloride current, we basically saw nothing. And only when we stimulated with a high concentration of acetylcholine did we get a sustained signal. If we put IP3 in again, it seemed sort of initially that it was just a very noisy record, and we were not really impressed by these short-lasting spikes. Now, luckily, very quickly, we were able to combine the calcium measurements with the electrophysiology. And it was Yuri Osipchuk who came to us from the Bokolomos Institute in Kiev, who for the first time did these combined recordings. And here now you see that when we actually don't see anything in the bulk cytosolic calcium concentration, we actually see very substantial rises in the calcium activated chloride current. And while these IP3 evoked fluctuations in calcium were not very impressive, when you look at the simultaneous electrophysiological recording, it's much more impressive. There's very, very clear changes in calcium activated chloride current. So it seemed that actually our conclusion in the 89 paper was correct. There were local changes in calcium, but they were just not very easy to see when we did what was really measurement of bulk cytosolic calcium concentration changes. Then we were able to do imaging a few years later, and of course this paper in Cell became far more cited than the original paper in 1919, although that actually was the conceptually important paper. Now, what also very often happens, and so while it is sometimes unpleasant to get a kind of feedback that maybe your results are not quite right, there is nothing that is nicer when you immediately see your own results confirmed by somebody else. And in the end of '92, I came to the University of Tokyo and gave a seminar there. And after my seminar, where I talked about the data that were not published at that point, so this is January or February '93, Haru Kasai came to me and said, well, I actually have very, very similar data. And it turned out, which is again another lesson, both of us, without knowing anything about the other person's work, had submitted our papers to Science. They have been promptly rejected by Science. And we then, again, without any consultation with each other, both sent it to Cell. It's a remarkable coincidence. And finally, they were published back-to-back, as you can see, in the same issue. And while we use slightly different ways of representing the data, you can easily see that these local calcium spikes was the result. So this was a very clear demonstration of these microdomains that then also finally in the epithelial cell world began to kind of become accepted. And one area where this became very important was in relation to the mitochondria. To Ole Petersen, who I had known from the early 90s, decided to look at a very specific phenomenon, namely how mitochondria could be activated by calcium rises in the cell. This had been a very controversial area, and the actually very brilliant British physiologist Peter Baker, a close associate of Alan Hodgkin, who unfortunately lived a very short life, at a meeting in New York, he put it very succinctly that he could not see how the mitochondria possibly could play any role in cellular regulation in relation to calcium metabolism, because the affinity for the calcium uptake mechanism in the mitochondria was very, very low, and it was very unlikely, at least that was thought at that time, that the cytosolic calcium concentration ever would get to those levels. So Tullio persuaded young molecular biologist Serino Risotto to create a construct that put a quarine, a photoprotein that is calcium sensitive, into the mitochondria. And when they did these measurements, you see the remarkable result that the calcium concentration in the mitochondria becomes much, much higher than in the cytosol. So Tullio reasoned correctly, as it turned out, that must mean that locally, close to the mitochondrial calcium uptake mechanisms, there were microdomains where the calcium concentration became very much higher than in the bulk cytosol. And later on, you see some very nice work that illustrates, we see here the outer mitochondrial membrane, the inner mitochondrial membrane, the ER. The contacts are very close in the cell. And this is now, of course, fully accepted. It's also actually a very nice title of the paper. I think it's one of the best titles of papers. It actually very succinctly says exactly what they found, which is actually quite satisfactory. Sadly, Ole Petersen died last year, which was very sad and was an enormous loss to the calcium community. We had a nice meeting celebrating his 70th birthday in Padua in 2019, and here he is seen with Tim Rink, who sadly also has passed away, who of course together with Roger Chen were the authors of the first papers that used the new fluorescent calcium indicators. A few months ago, I had the privilege of participating in a very dignified memorial event for Ole Petersen at the Accademia delle Lincei in Rome. So what about the mitochondria in the pancreas? In the pancreas, it turned out, and this was quite a bit of a surprise, that there's a very specific mitochondrial localization in the pancreas. And that's again a kind of surprising thing. I mean, there are tons of EM pictures of the pancreas because it has always been a major object for morphologists because it's an interesting structure. But somehow nobody had ever remarked on the distribution of mitochondria in the pancreas. And actually, this is a live microscopy, not dead microscopy. So here you see a single cell. You see the thymotin granules concentrated in this area. And with the mitotrack of green, you see that the main localization of the mitochondria is sitting just around the thymotin granules. So the mitochondria basically subdivide the cell into an apical part where the granules are and where the secretion control will occur, and into a basolateral part where the nucleus and the major part of the endoplasmic reticulum is sitting. And actually, this localization of the mitochondria is responsible for the local calcium signaling. So if you break down the electrical potential difference across the inner mitochondrial membrane, then you can see that these spikes, which are repetitive with IP3 infusion into the cell, become transformed into a global calcium wave. And actually, we don't have time to go into this today. This is actually pathology. This is exactly what happens in acute pancreatitis, where we get this sustained elevation of calcium that is dependent on continuous entry of calcium through the plasma membrane. And that will actually destroy the cell, and the cell will die in necrosis. So the physiology is here. The physiology is these short-lasting spikes, and each of these spikes give rise to a pulse of exocytosis. It also illustrates here the stimulus-metabolism coupling. You see here the calcium signal is captured as a change in the calcium-activated chloride current. We measure the calcium in the mitochondria, and we measure the Krebs cycle activity by NADH autofluorescence. So stimulus-metabolism couple is very important because we need to match energy production to energy need. And when the gland starts to secrete both enzymes and fluid, it needs more ATP. And that is actually organized by this calcium uptake into the mitochondria. Now to go back 50 years, and that will be the last bit of my talk. Very early on, this is work I did when I was on sabbatical in Cambridge in 71, 72, when we started working on the electrophysiology, and we demonstrated that both these two major stimulants of pancreatic enzyme secretion cause depolarization, and also that clearly that involves two separate receptors, because if you put atropine in, then of course the acetylcholine evoked depolarization disappears. But if you look at the CCK effect in the presence of atropine, it is completely unaffected. So there are clearly two separate receptors. And here we just see the earliest evidence for calcium release, followed quickly by enzyme secretion, and also the earliest evidence that this is not a neuron-type cell, because if you depolarize the membrane with a high potassium concentration, nothing changes in terms of calcium homeostasis. So this, in a sense, was generally accepted in the field, but then suddenly came a surprise that rumors began to circulate that maybe this was actually of no interest, the CCK effect, because human pancreatic Eisner cells don't have CCK receptors. I was rather suspicious of this finding, because all other species that we investigated, pigs, guinea pigs, mouse, rat, all had CCK receptors very clearly. But this paper published in Gastroenterology from a well-known group showed even a high concentration of CCK having no effect, and carbachol activating the muscarinic receptors, increasing the calcium. I was mainly suspicious of the paper because they had to use very high concentrations of carbachol to get a response, extremely high concentrations. So I suspected that these cells were maybe not in a good condition, and that many of the receptors had been digested off in the process of isolating the cells. So, of course, the only way to approach the problem is to do it yourself. It's not easy to work with human tissue, but we had a good collaboration in Liverpool with surgeons in the gastroenterology unit in the hospital, and we got fairly fresh human tissue. Pancreas is very difficult in this respect, because, as you know, the pancreas contains all the enzymes that are needed to digest all the food products that we eat, which means, of course, that it can also digest the pancreas itself very effectively and its surroundings. So it's actually not very easy to get, and even the time it takes to get the pancreas from the operating theater into the lab is already quite a dangerous moment. But nevertheless, we got a positive result with low picomolar CCK8 concentrations, which are the physiologically relevant concentration, and in the presence of atropine and tetrodotoxin, which really means that these responses are very unlikely to have been mediated by nerve endings that for some reason were adhering to the pancreatic Eisner cells. The controversy nevertheless continued, and as always in such cases, people say, yeah, one group says that there are CCK receptors, but another group says that there are no CCK receptors. But finally, another paper appeared from the group in Toronto, which actually also confirmed our data, and it turned out, as you can see here, that we did actually have very nice short-lasting spikes. So this is not a completely trivial problem, because one of the main processes, and Miklós Sahin-Tóth mentioned this earlier, of pancreatitis models is actually to hyperstimulate with CCK, or the frog peptide equivalent caroline. So if actually human cells don't have CCK receptors, then that would seem to be a particularly irrelevant way of doing it. And in fact, it is clear that human cells do have CCK receptors. And this is actually an extremely interesting phenomenon, which still is in search of a full explanation, because this must be the most extraordinary amplification system that I have heard of. CCK in concentrations as low as 1 to 2 picomolar can actually activate calcium signaling and secretion. If you try to make calculations on the number of CCK molecules that will be surrounding an Eisner cell at that kind of concentration, this is a very small number of molecules. So this amplification system is something quite extraordinary. I'm not aware of any other hormone that can induce effects at such extremely low concentrations. It also turned out that it's a completely different transduction mechanism. Initially, when people hadn't paid attention to the actual concentrations, and again shows the importance of doing concentration-response relationships, if you have a high concentration of CCK, you activate phospholipase C and generate IP3. But if you use low picomolar CCK concentrations, there's no IP3 formation. But another transmitter, nicotinic acid, adenine dinucleotide phosphate, is generated, releasing calcium from acid stores that then in turn release calcium from the endoplasmic reticulum. It's a rather complicated mechanism, and there are still quite a few elements there that are not totally clear, but it's very clear that it is a totally different transduction mechanism. And interestingly, the pattern of signaling is also not quite the same. So if you try to match the CCK concentration to the acetylcholine concentration that gives a particular frequency of short-lasting spikes, then as you can see, you will, in this case of CCK, see much broader global elevations of calcium. And why this is so, this is still actually not known at all. So there is still a bit of a mystery there, and I will kind of end with that mystery, acknowledge the people who did the work over very many years in my laboratory. There are many more people than that, but these are the people who did most of the important experiments. And in addition to the people who did the experiments, there are also very many people who influenced me very strongly. So you could call them influencers, friends, mentors. Some of them were younger than me, like Peter Hegge. Others were quite a bit older. But the interaction with these people, this is what has made life very enjoyable. At meetings, when you discuss with people, and then you get ideas, you get feedback to your ideas, bounce back on you. And I certainly hope that even in spite of my high age, that there will be further opportunities, and I certainly look forward to many further interactions with Peter, because that has always been a very enjoyable part of my life. So thank you very much for your attention.
Thank you very much, Ole, these fantastic presentations, and I really loved how you started with bad experiments. I remember I was once in Lindau at the major Nobel conference, and I am sorry, but at that time I was not sure is it important, but one of the laureates said that in his life, 50% of what he found is true and 50% is false. The only problem, he doesn't know at that moment which is false and which is true. So actually, students, questions?
So you said you were one of the founders of Academia Europaea. So my question would be that, did you had any serious motive behind that?
Sorry, I didn't quite catch. I have to say my hearing is not very good. Maybe.
The question was that you are a founding member of Academia Europaea. So did you have any purpose, direct purpose of when you realized that the importance and established the Academia Europaea?
Yeah, I mean, it was not, of course, me personally. I mean, I was invited by Sir Arnold Burgin, who at that time was the Foreign Secretary of the Royal Society, and who actually had the idea of creating the Academy. I was lucky enough at a relatively young age that he asked me to join the Academy at that particular point. His idea was that in the Europe that was emerging, in addition to the many excellent national academies, it would be sensible to have a Europe-wide academy. it was also becoming clear that the European Commission would not really be interested in receiving advice from one academy in one country and another academy from another country and yet another academy from a third country. That there was a need for a real European academy. However, for quite a long time, it did not actually work out like that. The European Commission did not seem to be particularly excited about the creation of the European Academy. And it was actually only our greatest president so far, Siert Klöting, who is very happy to see him in the audience today. And Siert Klöting, during his presidency, managed to present Academia Europaea as a very attractive organization for what was then the new scientific advice mechanism created by the European Commission. So in 2016-17, this scientific advice mechanism was actually created. And that, in a sense, was the fulfillment of our original dream of what Academia Europe could be. So there are other organizations that are involved in this scientific advice mechanism, but Academia Europaea is one of 5 all-encompassing European networks, but it is unique in the sense that unlike the other federations, it is an individual member academy. And Academia Europaea has actually played a major role. There's a further irony here, since we are sort of into what happens in strange ways during my life, that the UK, of course, who actually in this particular case is one of the very few cases where the UK actually did something positive for European integration. Then, of course, political changes occurred in the UK, and UK withdrew from. And now we are in the ironic situation that because Siert Klöting decided that it was the knowledge hub in Cardiff, of which I am honored to be a director, that should actually coordinate Academia Europaea's work for the European Commission, that we are now in the situation that the Academia Europaea's work in the scientific advice mechanism to the European Commission is currently financed by UKRI, so that's basically the UK Research Councils. So that's a quite ironic outcome that after all these tribulations, just now actually the UK itself directly pays for the scientific advice mechanism. So it just shows again how things very often develop in a completely different way from what you thought, but certainly Siert's vision, which reinforced the original vision of Arnold Bergen, has come to fruition, and we are very happy about this situation.
Thank you very much.
Thank you. Other questions? Yes, on the very back.
Do you think that your scientific achievements are majorly impacted by people you were amongst or were inspired by?
Okay, I'm sorry, my hearing is...
The question was that do you think that your the question was whether your scientific achievement, how much it was actually influenced by the people around you.
I think the influence from many of the people here was actually quite strong. I mean, obviously, the patch clamp work was, of course, by definition, very strongly influenced by the brilliant work of Vendégek and Bert Schekman. The work on the calcium signaling, I had very strong interactions with Mike Barrage, who I became very friendly with, and we met again and again and again. And Mike was a great person to discuss with because he was constantly making these diagrams again and again and again and trying to understand how things work together. So those interactions were very important, actually. And I think just to be able to present your findings at a meeting, get feedback, and in the discussion sense what other people think is important and what is not so important. These things are actually very, very helpful. So I would say a great deal, actually. I think my research work would have been very different if there had not been these interactions with many of these wonderful people.
Thank you.
Okay, thank you very much, and once again, thank you very much for everything what you have done for science, and we also would like to commemorate you with the plaque about your achievements. So please come to the middle, and I ask András Varró to deliver the certificate.